Okay, right, perfect. So, right. So, first part, before we start rushing through the important data, just to understand the whole organization. In males, you've got your testes. Then from your testes, you've got your duct systems, your efferent ducts, your epididymis, your vas deferens, coming the way ejaculatory ducts entering into the urethra. And you also have got something that you call your accessory glands, which you are your seminal vesicles, your prostate glands, your bulbourethral glands. Okay, so we just need to remember that. And the functions of the test is actually to produce gametes. Okay, so spermatozoa. So those are some of the functions of the testes, right? Okay, perfect, right? So yeah. Okay, this is not important. This is not important, right? So this is our testis, right? You find that the testis is going to be surrounded by a layer of dense connective tissue that you refer to as the tunica albuginea, right? Perfect. And we say that the tunica albuginea on the posterior aspect is going to be thickened to form something that you call your mediastinum testis. Done. But if you look closely again, you remember that your testis, right? What it does is, as it's descending down the abdomen to lie in the scrotum, it will do something that is to carry with it to carry with it it's like peritoneum, right? The peritoneum, the tunica vaginalis, right? And you're going to be having layers of the tunica vaginalis that are in direct contact with the testis that you call the visceral layer. And then your peripheral layer is not going to be in direct contact, it's going to be in direct contact with your scrotum, right there. So just to get that out of the way. Then right now the data, I think we spoke about, I think all of this you have spoken about it. So you find that from your tunica albuginea, you've got this septum coming in to divide the testis into around 250 lobes, okay? And then each lobe will have 1 to 4 seminiferous tubules, right? That you just need to remember. And then seminiferous tubules, like then in between seminiferous tubules, you're going to be finding your interstitial cells of Leydig, which are endocrine cells, right? Because they will be synthesizing your testosterone, right? We don't need to really go much into the histology as of right now for the exam. We just want to go to things that are pivotal, right? As of right now. There is something that you call, remember we said we have got 1 to 4 seminiferous tubules. If you want to discuss the structure of the wall of the seminiferous tubules, right, you are going to be having that, you realize that your seminiferous tubules are going to have your smooth muscle and everything, but they are not going to be having smooth muscle. They actually have something that you call myoid cells in connective tissue. That's the support substance of seminiferous tubules, right? And then the epithelium there is going to be two types of cells, right? The epithelium of seminiferous tubules is going to be Sertoli cells and your spermatogenic cells. So you can say the type of epithelial and seminiferous tubules, it's stratified. I think this one in our exam, right? So it's stratified epithelia because your spermatogenic cells are going to be like in varying stages of maturation, and they'll form up more than 4 or more layers of of 4 layers of cells, right? And then yeah, so that is that. So for now, good. We don't want these ones. So just this. So these are your Sertoli cells, and then these are the cells that you're talking about, right? The germinal epithelia, right? So if you look at it, you can actually see that you can say that the cells, they are stratified. And then if you find that you've got tight junction between these Sertoli cells and these Sertoli cells, there is a tight junction, right? And then everything that is this side, we call it adluminal compartment. Everything that is this side that's green, we call it basal compartment, right? So you find that because you form your primary, secondary, and tertiary, your primary and secondary spermatocytes after birth, your body will actually do what you call autoimmunity. Like they will, you to consider them foreign, and then it's going to go and attack them. So whenever they are formed, your primary spermatocytes, they will disrupt this blood testis barrier, and then and go to the abdominal compartment, and that's where development is going to continue. So your Sertoli cells protects compartment, that the things that are in the abdominal compartment, and that is any question? Okay, perfect. So but also remember that your blood testis barrier is the strongest barrier in the human body. It is the strongest barrier in the human body. Perfect. Mhm. Now we want to speak in detail about your Sertoli cells. So your Sertoli cells, they are there. One function of Sertoli cells that you should not forget, they are phagocytotic cells. They support your spermatocytes. They nourish them. And again, you said on lateral margins, they are joined by tight occluding junctions to each other, and then they will divide the basal and the abdominal compartments. Okay, we spoke about this, and you said the idea is to protect things in the abdominal compartment from autoimmunity, right? Still, again, this is the thing that you're talking about. Just see that those that your tight junction here separating these things and these things done. Okay, so that's your Sertoli cells, and you're speaking about the function. Yeah. Okay, so your again, lastly, your spermatogenic cells, right? We said they're going to be primary spermatocytes, secondary spermatocytes, your spermatids, and your spermatozoa, which will be occupying your abdominal compartment, and your spermatogonia are going to be occupying your basal compartment. Done. Right. So now, when you produce spermatozoa from the seminiferous tubules, they will start to move through straight tubules. They go straight tubules, then reed testes. From the reed testes, they go efferent ductus, epididymis, the vas deferens, and all the way to the ejaculatory duct. So that is the movement that they move through. But here now, things that you just need to remember is that the type of epithelia that lines all these areas, your type of epithelia and your support tissue that is there, and the muscles, like really the arrangement of the muscles there, that is what we really need to remember. So lastly, before we go there, we just need to speak about something else that you call your interstitial tissue, right? So the interstitial tissue is that's tissue between your seminiferous tubules. This is where you find your Leydig cells, okay? And they are the ones, this is the ones that are responsible for secretion of your what, your androgens. They're talking about your testosterone in males, right? And there in the interstitial tissue, that is where you find your fibroblasts, your mast cells, and everything. But one thing to remember is that your Leydig cells, they are rounded, have a central nucleus, and they are eosinophilic. Leydig cells, they are eosinophilic. Never forget this part, that they are eosinophilic, right? And then if you really want to speak about it, you have got the male reproductive system. Yes, got intratesticular ducts and extratesticular ducts. You have got your tuburecti, your testes, your ductus efferens, and yeah, so that is that. So what I want you to see, right here, perfect. You find that you start with your straight tube, right? You see, initially the straight tubules, they will be continuous with your seminiferous tube, right? Seminiferous tube, we spoke about it having, Sertoli cells and spermatogonia, right? So this tubule recti initially will also have Sertoli cells, and then they will move to have simple cuboidal epithelia and are supported by dense connective tissue. So this is what I said we need to focus on: support and the epithelia, right? And then when we move to the rich testes again, the epithelia continues as simple cuboidal. Okay, right? The epithelia continues as simple cuboidal, right? And your capsule is going to still, like, your support system is going to be connective tissue again. And then when you move from the rich testes to the efferent ductus, you start to find you have got two cells now, right? You have got your cuboidal. In this different ductus, you have got both cuboidal, simple cuboidal, and your what? You have got your simple cuboidal initially, and then you have got your ciliated columnar epithelia. So you have got these two cells. And then your support layer is going to be of secular smooth muscle, right? So that is for the intra genital ducts. I think I've got one diagram that I'm going to share with you at the end that diagram is important. We need to remember everything on that diagram. Then the male reproductive system, you've got what you call your excretory ducts, which are on the outside. You're starting from your epididymis, right, which is 6 meters long. So this one is now lined by pseudostratified columina, right? Pseudostratified columina, right? Perfect. And then you find it's going to be supported. You are going to be having smooth muscle array again in the epididymis, supporting your cells, right? And then understanding that in the epididymis, your cells, they are covered by stereocilia, long stereocilia, right? Perfect. So we find that you have got two types of cells in the epithelia of your epididymis. That means you have got rounded basal cells and columnar cells that have got this stereocilia. So that is that part. Then from your epididymis, you move to your vas deferens. Okay, so your vas deferens, things that you need to remember again, epithelia is still pseudostratified, and at the same time it's got the mucosa has got numerous longitudinal folds, and at the same time this one is the one that has got 3 layers of fibers: inner longitudinal, midsecular, and outer longitudinal layers of smooth muscle. This one you need to remember. Yeah, okay. So but then the epithelia is pseudo stratified. Done. Right. So with that we are done. I think lastly that we need to speak about, the efferent is not there, right? Your ejaculator duct is going to be lined by pseudostratified columnar and columnar epithelia. That you need to remember, right? Now we have got things that you call accessory glands. Okay, accessory glands of the thing is, we need to remember this one, right? So your of the accessory glands of the male reproductive system, the largest one is your prostate gland. Okay, and it is going to be pierced by the urethra, it's going to be pierced by your ejaculatory ducts, and this is how we divide it into your 5 lobes that we speak about when you're in gross, right? So you find again, it's going, the stroma of it is going to have got connective tissue and smooth muscles forming the stroma of the gland. Okay, so you've got connective tissue and smooth muscles, right? Right, perfect. So that you need to remember, that it's got smooth muscles as well, right? So again, your prostate is going to be having glands, right? So your glands are going to be compound tubular alveolar glands. I think if you read Janquera speaks about them being tubular acinar glands. Okay, but then if you read Defoe's, it's going to tell you that they are tubular alveolar glands. Now what you need to remember is you have got, in these glands we have got the mucosal glands, the submucosal glands, in the main glands of the prostate, right? So, and then you've got 3 regions: your central region, your transitional zone, and your peripheral zone. So the central zone is the region that is closest to the urethra, and it has got the shortest mucosal glands, right? And then the transitional zone is just outside the central zone, and has got submucosal glands. And then the largest zone, you find it as the peripheral zone. It's got the largest glands again, and it's got the main glands of the prostate. So things to remember about this is that benign, okay, this is just the thing. So this is the central zone, your transitional zone, and your outer peripheral zone. Things to remember is that your central zone is more prone to benign prostatic hyperplasia and compression of the urethra as compared to the outer part. So, and then the peripheral zone is more common to prostate cancer, which is an adenocarcinoma. Done. So this is that is just what you're speaking about. So you find that, well, you can have got what you call a, there's another process that can occur, right? Whereby you said Guti, the what you call it, the lumen of the tubular alveolar glands, right? Yes, got like what you call it, prosthetics consecretions, whereby you're saying your prostate is going to be secreting your glycoproteins, your and a lot of other substances, right? And then these are going to form what you call your corpora amylacea, they look like cysts, right? And then they are going to be increasing as a person grows in age, like they are calcified glycoproteins, right? But again, if you read your Janquera will tell you that these things, they are of no significance whatsoever, right? And then just to speak about prostatic secretions, okay, so this fluid is rich in lipids, your enzyme, yeah, and then um just that this idea that dihydrotestosterone is there to regulate secretions of prostatic glands. Okay, so that is that. Lastly, the most important thing, your seminal vesicles, they will secrete 70% of the ejaculate. This question was there in our exam. So majority of the secretion comes from the seminal vesicles, right? You have got two of them, right? And then you've got a folded mucosa. So your prostate gland and your seminal vesicles, they are both going to be lined by pseudostratified columnar epithelia or simple columnar epithelia. Okay, so that is done. So that is that done. And then your lamina propria is going to have got your elastic fibers, and you find that it's a highly tortuous tube. It's like bended, like it means like it's bended. Okay, in another way. And then you also find you have got folds that are located within your seminal vesicles, and then the idea that I said, it forms this number of secretions, right? So with this, we are done. But I think before we wrap up and I ask for questions, we just need to I just need to open like a very beautiful picture. Let me open a picture for you guys that I think is very important for us to appreciate before we move to anything else. What is that prayer that's written on your heart? Holy Ghost, expose me to it. What is that prayer? To read something, I think there is a very beautiful picture that I think we ought to also just read. If you are tired today and then you don't want to read, just go and open this tiny picture in Janquera. In Ganon. Sorry, in Janquera, yeah, in Janquera. Right? Let me just try to get it and post it on the group. Shift S, right? Perfect. Here it is. So that we are able to just appreciate something. So right. So yeah, if you guys want a 2 minute break, I think we can just take a break soon after. So I just want us to find some time to just appreciate this picture that I just posted, right? So this is just like a picture in Gano, in your Janque, right? Just go and appreciate this picture. So it tells you, yes, what you need to focus on. This seminiferous tubus, straight tubus, reed testes, efferent ductus, epididymis, vast difference, right? Just focus on the type of epithelia, which you say the seminiferous tube has got spermatogenic epithelia, Sertoli cells, and your germ cells, right? And we said spermatogenic epithelia is stratified. And we said that the support system of your seminiferous tube is going to be your myoid cells and loose connective tissue. And if you move on to your straight tube, yeah, your straight tube, we are going to find that it's going to be having what? Is going to it's like is the one that follows the seminiferous tube, right? So it's going to be having in the proximal segments, it's going to be having is going to be having your Sertoli cells, and as you go more distal, it's cuboidal and it's lined by connective tissue. But if you move to your reed testes, your reed testes, your reed testes is like just a continuation, so it's going to be lined by cuboidal epithelia. So I want you to just see that it's a sequence. We are moving from spermatogenic epithelia here. From spermatogenic, we move to cuboidal, and then as we move to the efferent ductus, moving more outside, you start to having cuboidal and columnar, and then as you move outside now into the ejaculator duct and your vast difference, you now have got pseudo stratified epithelia. That's just what I want us to appreciate, and just to know that the just know that for your epididymis, right? you have got pseudostratified with long stereocilia, like the cells have got long stereocilia, but when you go to the vas deferens, the stereocilia it's very few. So this is just the picture that I wanted us to appreciate. So we can, for those who want to take a break, I think before we do the MCQs, we can just take maybe a 2-minute break, as Kachepa has suggested, and then we'll just come back, throw in the MCQs, and then we just wrap up by the female reproductive. Is that good?
Yes, sir.
Okay, perfect. Okay, so our 2 minutes break is up. We can restart. Is everyone ready?
Yeah, 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 we are ready.
Okay, perfect, right? So what I last said was I was just speaking about this diagram here. This, I think we have all seen it. Those who have opened Janquera, what we just need to understand and appreciate is this: that yeah, from this one, I think we can just appreciate that the epithelia, what it does from your seminiferous tubus is just that it's like moves in a trend whereby you're saying your seminiferous tubus have got spermatogenic epithelia and acetylate cells. Straight tubus that follow the seminiferous tubus, we have on the initial part we have acetylate cells, and as you go more distally, you're going to be having cuboidal epithelia, simple cuboidal. And when you move to your root testis, it's going to be simple cuboidal. And then as you move from your root testis to your efferent ductus, you're going to be having simple cuboidal alternating with simple columnar ciliated epithelia. And as you move from there, you're going into your external duct, that's your epididymis, your vas deferens. You have epithelia which is pseudo stratified columnar with stereocilia. And I said that more of these stereocilia, you find them in your epididymis inside of you. System for the seminiferous tube is your myoid tissue, and then your straight tubes you find connective tissue, and your root testis is specifically dense, irregular. And as you move outward towards your efferent ductus, you find that's when you start to form your thin cells, thin cellular layer of smooth muscle. Then your efferent ductus, your epididymis is going to having secular smooth muscle initially, and then inner and outer longitudinal layers in the tail. Then when you move to your vas deferens, that's where you've got your three layers: your inner longitudinal, midsecular, and your outer longitudinal. Just if you are able to appreciate this diagram for male, and then appreciate that your tunica albigonia is just that it's a dense connective tissue, and then appreciate that your Leydig cells, they are eosinophilic, and the function of your stellar cells your to divide your things into a basal compartment and your abdominal compartment, whereby things in the abdominal compartment, they are protected from your immune system, right? That means your primary spermatocytes, secondary, and so on. But your spermatogonia, they're in the basal compartment, there is no protection. And that when you form primary spermatocytes, they will disrupt that blood testis barrier, which is the largest barrier in the human body, which is like the strongest barrier in the human body, and they will disrupt it to enter into the abdominal compartment. But even when they enter, they are still adhering to your Sertoli cells. These are the nuggets that you think you need to be able to remember. So lastly, to be able to solidify our information, we just need to go through a few MCQs, right? So MCQ number 1: concerning the male reproductive system, Leydig cells produce structural and nutritional support to the developing spermatocytes. This is false because this is the function of your what? Sertoli cells. The tight junctions between Sertoli cells form blood testis barrier. This is true. And then both epididymis and vas deferens are lined by the same type of epithelia. This is true. It's pseudostratified with stereocilia. Then stroma of the prostate glands is rich in smooth muscle cells. I remember I remember I mentioned this over and over that your prostate gland has got smooth muscle cells. Prostate gland has got smooth muscle cells. Also, one thing that I forgot to mention while you're doing the presentation is that you have something that you call your bulbourethral glands, which are homologous to your greater vestibular glands in females. What they do is they just create something that lubricates the penile urethra in preparation for intercourse. Yeah. I'd forgotten that part. Now, lastly, the glands of the peripheral zone of the prostate are prone to benign prostatic hyperplasia, falls the peripheral zone is more prone to an adenocarcinoma, cancer of the prostate. Right. Now, and then next, if we move to histology of the testis, it's covered by loose connective tissue capsule. I think we have already discussed this, and you said this is false. Why? Because it's a dense connective tissue capsule, the tunica albuginea. Then adjacent Stelli cells are joined by tight junction forming blood testis barrier. True. Then developing spermatocytes are located in the abdominal compartment. I just mentioned this now. True. Your Stelli cells are phagocytic. We mentioned this. True. Common testicular cancer commonly involve Leydig cells. False. They commonly involve the spermatogenic, your germ cells, your spermatogenic cells, right? Then regarding male ducts and accessory glands, cuboidal epithelium lines the efferent ductules. This is exactly why I was mentioning that we need to be able to remember this diagram that I was just mentioning, that efferent ductules, we have alternating pages of simple cuboidal and simple columnar ciliated epithelia. Okay, back to our MCQ. Right. So yeah, you just answered that part. Yeah, so true. Then smooth muscle cells are absent in the walls of the epididymis. False. Remember that in the epididymis you actually have got, there are actually two layers actually. You remember initially you've got a circular layer, and then as you go distally, inner and outer longitudinal layers. Then you see the questions that are being asked now, the type of epithelia that lines these organs. So you just need to go and appreciate that diagram that I just showed you. Then the epithelium lining the seminal vesicles is glandular. So this is not mentioned directly, but from all the sources that I've looked up, this is false. The epithelium is not glandular epithelia. The pseudostratified epithelia lines the urethra. Well, we did not mention this because this is part of the urinary system, but yeah, and we mentioned this when you're doing embryology actually. So this is true. Then the cancer of the prostate is called an adenocarcinoma. I think I was just mentioning it like it's an adenocarcinoma. Then right, development of the male reproductive system. In the terminal enlarged portion of the terminal enlarged portion of the penile urethra is lined by stratified squamous epithelia. Well, this is true, but we didn't mention it here. We haven't mentioned because we haven't done the urinary system. But just there's that enlarged portion before you exit that narrowest portion, which is your meatus. Remember that the sponge urethra comes from the caudal portion of your urogenital sinus, but the most distal portion of the O is going to come from your ectoderm, right? That invaginates inside the thingy. But then the blood testis barrier is formed from the zonal occludence tight junctions. Done. Then Leydig cells have basophilic. I remember I mentioned that you should never forget that it's eosinophilic. Then your vast difference is lined by pseudostratified columnar epithelia with stereocilia. True, but the stereocilia, need to remember that they are more in the epididymis. Then central zone of the prostate gland is more prone to benign prostatic hyperplasia than peripheral zone. This is true. Remember, I mentioned this actually. The peripheral zone is more prone to adenocarcinomas or prostate cancers. Then, yeah, I think this one we can just mention two things that are important to us. The muscularis is more prominent in the vas deferens than in the epididymis. This is definitely true. In the vas deferens, you've got three layers, then unlike in your epididymis. When the blood testis barrier protects cells of the basal compartment of the seminiferous tube, it protects cells that are in the what? In the abdominal compartment. Then I think this one is getting answers. This is our last one. The prostate gland epithelium is stratified cuboidal. the prosthetic glandular epithelia stratified cuboidal. Remember, I mentioned that the type of epithelia there is the same type of epithelia in the seminal vesicles, which are your what, pseudostratified columnar or simple columnar. Then your glandular epithelial elements of the prostate are lined by ciliated columnar epithelia. False, your glandular elements, they are not actually lined by this. It's actually pseudostratified. But yeah, and then next, the height of seminal some of these sections, it's actually not part of Janquera, part of T-Force. I actually wanted to skip this MCQ, but let's just do it just in case. The seminal vesicles exhibit marked folding. Remember, I said with the mucosa has got numerous folds of the seminal vesicles. This is true. And then your bulbourethral glands are mucus secreting compound tubular alveolar glands. This is true. I think this is just like a statement that was taken directly from Janquera. So oh yeah, this is the last one. Regarding the male reproductive system, the prostatic urethra is lined by transitional epithelia. No, false. The prostatic urethra is lined by what? By our at least that's this. What is this epithelia? Our pseudostratified epithelia. Then if you read, actually, if you let me just show you the thingy. If you open Janquera, I think because I think we have just noticed that they keep on asking us about this epithelia. I think just let me just show you the picture of this. But it's actually part of the development of the urinary system, which if we are listening to Dr. Show, it's not that important for our exam, but just so that we have a complete knowledge, especially on this thing about the urethra. Let me just get to it. Urethra. Okay, I cannot project my slide, but just this idea, like we have got the prosthetic urethra, the membranous urethra, and your spongy urethra, right? So your prosthetic urethra is going to be like 4 centimeters long and is the one that passes through the prostate gland, and it's actually lined by our urothelium, which is actually transitional epithelia. Then our membranous urethra is going to be the one that is a very short segment. Remember, it passes through the external sphincter, and then it's actually lined by stratified columnar and pseudostratified epithelia. And then the spongy urethra, the longer portion, is going to be lined again by stratified columnar and pseudostratified columnar epithelia. And as you go more distally, you've got stratified squamous epithelia. Okay, so far so good, right? So our prosthetic urethra is going to be having is going to be lined by our urothelium. Our membranous urethra is going to be lined by stratified columnar and pseudostratified epithelia. And then the sponge urethra, same again, is going to be stratified columnar and pseudostratified epithelia. So these are the kinds of epithelia that lines our urethra. So I think I will, since I cannot project it, let me just try and create a slide here and then try to control V. Yeah. So yeah. So this is just like a section from Ganong that I was just using. So from Janquera. Okay, so it just tells us about it. I think we can just have time to just go and look at it. It says prostatic urethra from urothelium, then membranous urethra, short segment from the external sphincter, and then it's lined by stratified columnar and pseudostratified. Yeah. Okay. Lastly, so prostatic urethra by transitional. So transitional is the same as the urothelium, and then The vast difference has got a thin muscular wall. This is false. It's actually thick, 3 layers actually. Then cytolith cells are usually found in the interstitial tissue between seminiferous tubules. No, these are Leydig cells that are being described. Then the mucosa of the seminal vesicles is lined by pseudostratified columnar epithelia. True. Okay, so with this, we are done with the development of the male reproductive system. Anyone with any question before we move? Okay, so there's no one with any question? Right. So I think in the next 15 minutes, we will try to see if we can cover through the female reproductive system, which apparently is the hardest. It's not the hardest, but it's